Hello everybody. In our last lecture regarding Salmonella, we mainly talked about the pathogenic Salmonella strains that cause, say, typhoid fever or gastroenteritis. Our primary focus was on the molecular mechanisms that these bacteria use to generate their virulence. So in our continuing discussion today, we will talk about the emergence of multi-drug resistant pathogenic Salmonella strains called superbugs. Okay. During the last decade or so, all over the world, many studies have reported emergence of some Salmonella pathogenic strains in the clinical context that are resistant to virtually almost all known classes of antibiotics. And thereby they are virtually untreatable. The disease caused by this pathogen, they are untreatable. Consequently, they are now included as superbugs. It has been found that these bacteria, they acquire their multi-drug resistant property by acquisition of many antibiotic resistance genes via horizontal gene transfer. And many of these, almost all of these horizontal gene transfer has been found to be occurring within the anthropogenic environment. Now, this acquisition of so many different antibiotic genes via horizontal gene transfer are extremely rapid, and that's why their emergence is like a surprise to the medical people. Now, there are certain genetic elements, DNA elements, called integrons that has been found to be the major player of imparting this type of multi-drug resistant property towards these Salmonella superbugs. Today we will mainly focus on the roles of integrons in Salmonella to generate these superbug strains, and also we will try to discuss something about how to counteract this global threat. So integrons, they are basically genetic assembly platforms that can promote acquisition as well as expression of certain DNA elements that remain embedded within the integrons, called gene cassettes or GCs in short. So this is a overall view of a general integron, I can say. These GCs, the gene cassettes, here only 2 gene cassettes are shown, but it can be many more in different integrons. In the 5' side, there is this functional platform of integrons. This is a conserved sequence. It's called 5' conserved sequence of integrons. We can see there are 4 critical components of this. One is the int-I gene. Then we have this at-I site, which is a site for DNA recombination in a specific manner. Then we have 2 promoters. One is PC directed towards this direction. Another is p-int directed towards this direction. Now, this int-I gene, it actually encodes an integrase enzyme belonging to the integrase family. It's basically a site-specific tyrosine recombinase. The main active site player is a tyrosine residue. This is expressed via this p-int promoter. Now, these gene cassettes, they contain open reading frames, and in many cases these open reading frames, they encode information for various different types of antibiotic resistance gene. All of the gene cassettes are coupled with a 59 base pair DNA element called at-C site. Okay, here for GC1 it's at-C1, GC2 it's at-C2. This is the site that actually take part in the recombination with the at-I site, and that recombination for incorporation of these gene cassettes from the environment are catalyzed by the int-I enzyme. This int-I enzyme sometimes also catalyze not only the incorporation but also the excision of gene cassettes. So here we have a generalized scheme of integration and excision of a gene cassette. Say this is already existing in integron. This is a 5' conserved part, and this is the already inserted gene cassette. Then incoming gene cassette is there, a new gene cassette. Via this at C site, it can recombine with the at I site via this catalyzed by this int I protein, and then it gets inserted here. You can see that when this is inserted here, the previous one is displaced towards the right hand side. And the int I protein actually catalyzes a reversible reaction, that is, it can also excise out this inserted gene cassette to its free form. When the gene cassettes are embedded within the integron, you can see that they are existing in a linearized form. But when they are free in the environment, they exist as a covalently closed circular DNA element. Now, these gene cassettes that contain the open reading frames, which it can most frequently be antibiotic resistant genes, they themselves do not contain any promoter. Consequently, they are not expressed until and unless they get help from another promoter. And that is provided by this PC that is already there within the 5' conserved sequence. This PC is a very strong promoter, and this system behaves like an operon because these gene cassettes are successively arranged. So this PC will express all these gene cassettes. I mean, it can be many more. Here only 2 are shown. So just like operon system, the gene cassette closest to the PC will express its ORF at the maximum level. That is one important criteria of the system. For example, both contain different antibiotic resistance genes. Then the antibiotic resistance genes in this ORF will be expressed at a much higher level than that in the next gene cassette. Now, obviously, there are 2 types of recombination going on, both site specific. For example, if the at I site and the at C site recombines, then we have insertion of an incoming GC. Like here, say in this integron, here is GC1 is GC2. Now a new integron is coming, GC3, with its at C3 site. When this at I site recombines with the at C3 site catalyzed by int I, then this GC3 will be inserted here. Okay, and see, the funny thing is that when GC3 is inserted here, GC1, which was closest to PC, is now moved downstream. On the other hand, recombination can also occur between 2 at C sites that leads to excision of a GC. For example, here is shown that a recombination between at C3 and at C1, again catalyzed by the int I protein, will lead to excision of GC1 in its circularized form.
Sir, the position of a gene cassette within the integron with respect to its PC promoter is variable. Does this have any special significance?
Yeah, the positions, they are variable with respect to the promoter PC, and that has a profound significance. Actually, that is one of the most intriguing beauty of this integron system. For example, here you can see that if this gene cassette is encoding antibiotic resistance genes against antibiotic 1 called ANTR1, this is ANTR2 against antibiotic 2, then obviously in this scenario, ANTR1 will be expressed at a much higher level than ANTR2. So this cell containing this integron will be more resistant to antibiotic 1 than antibiotic 2. Now when gene cassette containing ANTR3 is coming, recombining and getting inserted here, now the cell becomes more resistant to antibiotic 3 rather than antibiotic 1. Okay, so it's shuffling its position. Again, it can so happen that, say here you have antibiotic ANTR2, it can get excised out, and then again it can be reinserted here by recombination with at I. Then in that case, ANTR2 will be highest expressed than ANTR3 or ANTR1. This indicates that the position of the antibiotic resistance genes are always getting shuffled, and it's a dynamic situation. So in a population of cell, you will have different subpopulations. Say in one subpopulation will be more resistant to antibiotic 1, another will be for antibiotic 2, another will be for antibiotic 3, and so on. So here only 3 antibiotic genes are shown. The integrons can have up to 10 antibiotic resistant genes. So there will be always a type of subpopulation which will be resistant to a certain type of antibiotic. So whatever be the antibiotic challenge, the population is ready. Because say if you have added antibiotic X, then a certain population where the resistant gene against antibiotic X is most highly expressed will survive. The rest will die. But this population that has survived will then eventually proliferate and regenerate the population. So this is how by this special shuffling mechanism of integrons, the population uses a sort of trial and error to face almost any type of antibiotic challenge. The population is ready. Depending on the genomic location of integrons, Salmonella can have like 2 types of integrons. For example, one is called chromosomal integrons called CIs, another is called mobile integrons called MIs. CIs in Salmonella, they are located on the chromosome, say in the Salmonella genomic island 1 or SGI1. This is one of the hotspot to accommodate different chromosomal integrons in Salmonella. Some of these CIs in Salmonella are also called superintegrons because they can carry up to 200 gene cassettes, which may encode different metabolic functions or maybe unknown functions that we do not know of, and obviously some antibiotic resistant genes. Mobile integrons, on the other hand, though the name suggests they are mobile, they are not transmissible by themselves. They need help of other mobile genetic elements to move from one position to another or from one cell to another. One scheme is like this. Say you have a gene cassette here containing the antibiotic resistance genes. Then it is getting incorporated within the integron. The integron in turn is getting incorporated within a transposon. And this transposon then getting incorporated within a conjugative plasmid. You know that conjugative plasmid can transfer from one cell to another by the process of conjugation. So when it does so, it will carry the integron as well as the antibiotic resistance gene with it, and thereby the integron mediated antibiotic resistance will transfer from one cell to another. Now mobile integrons, they can contain up to about 10 gene cassettes, which in Salmonella usually encodes different types of antibiotic resistant determinants. That is why Salmonella mobile integrons are sometimes called resistant integrons or RIs, or even better, multidrug resistant integrons or MRIs. Now, based on the amino acid sequence of the N-type protein, about 5 classes of class 1 MIs have been reported, out of which class 1 MIs are the most common in the multidrug resistant Salmonella. That is why these class 1 MIs in Salmonella are one of the most well studied integron systems. They can carry so many different types of antibiotic resistant genes, and they are usually isolated from Salmonella strains of clinical interest from humans as well as animals. Usually they are associated with transposon TN21 or its derivatives. And these transposons are again embedded within some conjugative plasmids of Salmonella, allowing their efficient and rapid dispersion. Intriguingly, this INT-I1 gene sequence, that is for the class 1 MI in Salmonella, they are highly conserved from clinical isolates, but show some variation from other environmental isolates. So we have seen the 5' conserved sequence of Salmonella integrons. We have seen the so many different antibiotic gene cassettes which are shuffling around within Salmonella integrons. Now, class 1 MIs in Salmonella also contains a 3' conserved segment, and that's also very interesting. Say here, you have one reading frame encoding QACE delta 1 gene. Okay, so this encodes a deleted version of QACE, which confers resistant against quaternary ammonium compounds. We know that most of the disinfectants and antiseptics that we use, they are quaternary ammonium compounds. So a Salmonella containing this will become resistant against those antiseptics as well. Then following it is SAL1 gene. This confers resistance against sulfonamide group of antibiotic. So these two are as operons and they are expressed via this promoter. And then you have another promoter under which there is ORF5. This is of unknown function. We still have not yet been able to understand its function. Different antibiotic resistant genes belonging to the gene cassettes in Salmonella class 1 MIs, they include almost all antibiotic families. For example, beta-lactams, aminoglycosides, trimethoprim, chloramphenicol, phosphomycin, macrolides, lincosamides, rifampicin, quinolones, and even more. You name it. Whatever the modern antibiotics we use today, Salmonella will have a resistant gene within the integron. This, in combination with the resistant against quaternary ammonium compounds as well as sulfonamide antibiotics, makes Salmonella bearing this integron a versatile fighter against almost any sorts of antimicrobial challenge. Now, this antibiotic pressure created by different human context, they probably played a very important role in the selection and dissemination of these integrons in Salmonella. Because studies indicate that these integrons are most prevalent in Salmonella strains that are subjected to antibiotic pressure in different anthropological environment. For example, clinical or agricultural or poultry or animal farm settings. So this outlines different routes, general routes that lead to the dissemination of this superbug Salmonella in the environment from anthropogenic source and vice versa. For example, WWTP, that is wastewater treatment plant, they act as the main reservoir of these superbugs from various anthropogenic sources. It can be a regular human population, it can be hospitals, it can be industrial facilities, it can be antibiotic facilities, it can be slaughterhouse. So from here they accumulate in the plant. From the plant it can go to land farming, and from land farming to soil, and finally from soil to river. It can directly go from the plant to the river, as well as fish farms can contribute directly to the river. Okay. When they are in the river, they can easily go to the drinking source, and from the drinking source it can again reinfect the anthropogenic environment, and from the river it can go to the sea via estuaries, where it is now exposed to the environment in a much broader context. Now, the regulation of Salmonella integron activity has intrigued microbiologists for last few years. It has been found that it is induced by SOS response. Normally, SOS response is a global regulatory network. A bunch of genes are expressed which are under the regulation of the transcription repressor LEX-A, and that is induced by a certain type of stress that ultimately lead to DNA damage. Interestingly, it has recently been found that many widely used antibiotics such as fluoroquinolones, beta-lactams, trimethoprim, aminoglycosides, and others, when they are administered within bacteria, they generate DNA damage, okay? Mostly via free radical induced mechanisms. Now, these type of DNA damage can also activate SOS response. That in turn, by some unknown mechanism, can turn on the integrase expression. So now the integrases will be overexpressed, which in turn will raise the different recombination event of the gene cassettes containing the antibiotic resistance genes, and that ultimately will increase the probability of emergence of multidrug resistant superbug.
Sir, is it then possible to design drugs against the integrase protein to inhibit the integron activity?
Exactly. See, integrase are now being tried as a possible drug target, because we now know from this discussion that the integrase activity is at the heart of emergence of multidrug resistant superbug Salmonella. So if people can design and synthesize a drug against this integrase protein encoded by the integrons in Salmonella, then probably people will be able to block the activity. And that's what people are trying to do. But for that, we need more knowledge about the three-dimensional structure of Salmonella integrase encoded by the integrons. Here, a data has been shown where they have solved a structure where you can see the monomers have assembled as a tetramer, the monomers of the integrase INTI protein. In complex with the DNA, these ribbons are the DNA that are being recombined by site-specific tyrosine recombinase action of these tetrameric INTI protein. So more and more data are coming regarding structures of these integrase protein from different Salmonella strains, and it's quite promising that in near future we will have a good drug that will be able to inhibit this INTI integrase activity in Salmonella, so that we will be able to specifically block the integron mediated emergence of multidrug resistant Salmonella superbug. Okay. So that kind of concludes our discussion today. So in retrospect, we can say that we have talked about the emergence of a Salmonella which is virtually resistant to all sorts of antibiotics, and it's a matter of global concern because it is being generated all over the world. In Southeast Asia, including our country, there are so many cases reported every day, and even in the developed countries like Europe and United States, this is being a matter of profound concern for the last decade or so. Even they cannot prevent the emergence. So people are trying to understand the mechanism of this emergence, and that has been traced back to the presence of integrons and their activity. And we have seen that integrons are versatile reservoir of antibiotic resistant genes by which a population is prepared for any type of antibiotic challenge at any time. And we have understood some of the mechanisms, still a lot work to be done. And finally, people are thinking of a strategy to design a drug that will be able to block the integron activity and thereby prevent the emergence of these Salmonella superbugs. Thank you.